Hey there, this is Kathy Go again. I am back talking to you more about this lecture that's coming up. So let's begin with objective 4. So now we're diving into the inflammatory response. So one of the constituents of the inflammatory response are the plasma protein systems of the body. So one of the plasma protein systems is complement. Which consists of about 30 plasma proteins working together to augment the inflammatory response. They circulate in an active form, so they're not attacking things all the time. When they're activated, they trigger the complement cascade. So the major functions of the complement system include enhanced phagocytosis, increased vascular permeability, chemotaxis, and cellular lysis. So this is kind of a simplified version of what it looks like. So you have a cell that gets attacked by a pathogen. That cell then sends out signals which attract white blood cells. Macrophages is what's in this picture. The macrophage, or then the complement cascade destroys the cell contents, and then the stuff that's left floating around causes a clustering of bacteria. So that's sort of a big picture look. So, the complement cascade, in summary, results in opsonization, chemotaxis, and cell lysis, and agglutination. It works with the adaptive and innate immune responses. So you can see that this is a complex pathway, and there's different ways that it can be triggered. And those different triggering mechanisms are based on different types of pathogens, whether it's a bacteria, whether it's a certain type of bacteria. But they all wind up they come together at C3, and then the different products of that reaction result in opsonization, the membrane attack complex, and the results of chemical signaling, which result in vasodilation, increased capillary permeability, and chemotaxis. So the different main constituents that are responsible for these responses are the ones that you see there, and that happens at different points in the cascade. So the main effects of the complement cascade include opsonization, and that's when this invading cell is coated, and it creates chemical handles so that the bacteria or other parts of the inflammatory system can identify and grab onto these cells. The final common pathway that results in the membrane attack complex is C5b, and that just basically drills holes in the cell membrane of the pathogen and makes it blow up. And then when all these contents are out in the system, it enhances the inflammatory response through histamine, chemotaxis, and that part of the complement cascade is undertaken by C5a. So this, in terms of pathophysiology, one of the main effects that you can see is on autoimmune diseases. This is kind of the site of action where autoimmune responses happen, like in rheumatoid arthritis or lupus. So another protein system includes the clotting cascade, and that results in hemostasis. So the main actions of the clotting cascade include, there's an injury, there's brief vasoconstriction, that's to keep you from bleeding to death. Then a platelet plug is formed. Then there's blood clotting that happens. So there's, again, like with the complement cascade, there's different ways that this cascade can be triggered. So there's the intrinsic pathway, which is triggered by factors within the blood, and there's the extrinsic pathway, which is triggered by factors outside the blood. And so then you don't want clots to happen all the time. So it has a way of stopping this cascade and getting rid of the clots. And that's through fibrinolysis and clot retraction. So these cascades not only have to get triggered, but they also have to be turned off. So let's look a little bit more at the clotting cascade. So the clot formation happens in 3 steps. You get activation, where receptors are turned on, and that triggers the cascade. Then you get adhesion, which is where the constituents of the clotting mechanism, mainly platelets, stick to the injured endothelium. And then you get aggregation, which is clumping and forms receptor bridges. So you get primary hemostasis. Then you have the platelets, which trigger clotting factors, which then in turn trigger fibrin to be laid down, and that's kind of what makes the platelet plug. And then the plug is in place, and then these connections happen to kind of glue everything together. So then the third protein system is the kinin system. Now these are all very complicated, and I'm trying to kind of hit the main points here. So the main functions of the kinin system are to control blood pressure, inflammation, coagulation, and pain. So the effects of the kinin system are vasodilation, increased capillary permeability, and it works with prostaglandins to cause the pain response. So this is kind of a simplified view of the kinin cascade. So you can see that there are these precursors, then it's converted to, splits off, and then is converted to bradykinin, which is the active agent that causes all the pain response, and plasminogen plays a role in triggering plasmin relief. So the point is that it's related to the coagulation process. They're kind of integral. So that is that one. So another major constituent of the inflammatory process are prostaglandins, and they play a big role. Oh, I wanted to back up and say on the kinins, they're a powerful vasodilator. So when kinins really get rolling, they can cause shock because the blood vessels dilate, and then blood pressure isn't supported, and then people have septic shock. So that was backtrack. Now we're on to prostaglandins. So now how this works is, following an injury, arachidonic acid is converted into prostaglandins, thromboxane, and leukotrienes. It's considered a pro-inflammatory set of chemicals, and they're very potent vasodilators. So what happens is the cell membrane is disturbed, then arachidonic acid is released, and that triggers the COX pathway, which results in the release of prostaglandins and thromboxane, and also the lipoxygenase pathway, which results in leukotrienes. So you can see here this schematic illustrates that how medications can interrupt this process. So that's kind of interesting. So that's another constituent. So let's move on to objective 5, the cellular components of the inflammatory response. So there are different types of cells that participate in the immune response, which we talked about. So this is the adaptive immune response or the inflammatory response. So there's endothelial cells, platelets, phagocytes, and other leukocytes. So the main players in the phagocyte category are neutrophils, monocytes, macrophages, dendritic cells, and natural killer cells. And so they're active phagocytes. And the endothelial cells play a big role in the inflammatory response and clotting, as we've kind of discussed. They're the ones that play a role in triggering some of these protein systems that we were looking at earlier. Platelets, of course, play a big role in the clotting process and they also secrete chemical messengers which play a role in inflammation. And then there's other leukocytes that participate, basophils, eosinophils, and mast cells, and they play a role in releasing other chemical messengers, including histamine and other pro-inflammatory chemicals. And what they do is they carry these constituents around, and when they degranulate, they release all these chemicals into the intracellular and extracellular fluid, and that's what attracts cells and brings about inflammation and triggers some of these cascades, and they play a big role. So let's talk a little bit about the phagocytes. So there's different ones. You could consider neutrophils as the first responders in this process, and they're big phagocytes. They trigger a respiratory burst. They also opsonize pathogens. They release toxic chemicals like defensins, which then poke a hole in the pathogen. Macrophages, that means big eaters, and they're the cleaning crew. They play a role. They are also huge phagocytes. They also opsonize. And they make the handles for the phagocytes to hang on to. They put the handles on the pathogen so that the phagocytes can grab onto them. Natural killer cells could be like the police. They kind of patrol, they identify the major histocompatibility complex. They trigger apoptosis. And then dendritic cells are like the patrol. They also are just floating around in the system. They are big phagocytes, and they are antigen presenters. So they kind of cross over into the adaptive immune response. So the cells of the innate immune system, some characteristics are they use receptors to recognize foreign bodies. The cells work together through chemical communicators, cytokines or chemokines. The chemical messengers that are out there communicating between these cells can be pro-inflammatory or anti-inflammatory. So they're both kinds. Some examples of cytokines are interferons, interleukins, tumor necrosing factor alpha. So those are some pro-inflammatory cytokines. So let's talk about the cellular and vascular response. All right. So back to sort of the inflammatory response in general, you can divide the inflammatory response into the vascular response and the cellular response. Then, as a consequence, there's a formation of exudate and ultimately healing. So this is what the vascular response looks like. You get transient vasoconstriction. So this is all kind of carried out by the endothelial cells on the capillary level, where at the site of tissue injury. then the vessels dilate, resulting in hyperemia, which is just a lot of red blood cells in the tissues. This increases then there's increased capillary permeability. Then there's movement of fluid from the capillaries into the tissue spaces. This serous fluid plus plasma proteins draw more fluid into the tissues, which then results in plasma proteins trigger fibrin to support the clot. So you can see how all these systems play together. So in general, the chemical mediators of the vascular response cause increased capillary permeability, and they facilitate movement from the capillaries into the tissues. So initially, the serous, the exudate that's formed, this fluid, is serous. And it's mostly made of water and plasma proteins, like albumin is an example of a plasma protein that's in there, which then increases the oncotic pressure in the extravascular space, well, out in the tissues, and that sucks more fluid out there. So that's why you get so much swelling. So let's talk about the cellular response. So blood flow, so when there's swelling, the blood flow slows down. There's more blood in the blood vessels. Things slow down because these vessels are dilated, which then results in the white cells that are just out there in the circulation moving to the outside of the blood endothelial wall, up to the endothelial wall. And then they are drawn to the site of injury by chemotaxis, which is triggered by a bunch of different things. One of the examples of the white blood cells that are along the side of the blood vessel wall are neutrophils. They are short-lived and they're the first responders. They get into the tissue, they fight the infection, they die, and they form pus. And then monocytes are kind of the second wave, which then become macrophages, which are the cleaning crew, and they prepare for healing. And then later, the lymphocytes arrive, and they trigger the adaptive immune response. So this is kind of a big picture look at who does what in the inflammatory response. These different chemical mediators that cause a fever are interleukins, prostaglandin, E2. And what they do is they change the thermostatic set point by acting on the hypothalamus and the autonomic nervous system. The redness is caused by histamines, kinins, and prostaglandins, and that's because there's more blood flow to the area, and the vessels are dilated, and that vasodilation and hyperemia causes redness. Edema is caused by histamines, those vasodilators, kinins, and prostaglandins. That results in swelling. And that's caused by increased capillary permeability and fluid shifts into the tissues. And leukocytosis, which is increase in white blood cell count, because the thing that impacts or triggers that is chemotactic factors. And remember, chemotaxis is that tractor beam that draws the white blood cells to the site of injury. And that triggers an increased release of neutrophils and monocytes in the bone marrow. So this is kind of what it looks like. So one of the pieces of the cellular response is that as the blood vessel slows, the white cells go to the edge of the blood vessel, they kind of roll up, and then they stick to the edge of the blood vessel. Then they find these tight junctions, and they make a little foot, and they crawl through the space, and that's how they get out to the injury area. On the vascular side, there's arteriolar changes, there are venular changes. You get vasodilation, increased permeability, which results in redness and swelling. So one thing that is good to know about is a laboratory marker for inflammation and infection. So as we noted, neutrophils hit the site first, and so there's a whole bunch of them that are released from the bone marrow, and they're pretty short-lived, and eventually the body runs out of neutrophils. So when that happens, the bone marrow releases immature neutrophils, which are called bands. And then because on a standard lab form, the bands are to the left of the neutrophils, there is a relative increase of bands compared to neutrophils, and that's called the left shift. So you might hear people talking about that. That's what that's about. So this is what the vascular response looks like. And this is what the cellular response looks like. So you can see the margination and rolling up, and then the diapedesis, and then you got the tractor beam drawing the white cells to the site of injury. So let's summarize the main events of the vascular and cellular response. Vascular response is responsible for initial vasoconstriction, then vasodilation, increased permeability, and formation of exudate. The cellular response is responsible for margination, diapedesis, chemotaxis, and the release of pro-inflammatory chemicals. So, some pro-inflammatory chemicals are these, histamine, serotonin. So histamine and serotonin come from the degranulation of mast cells, and they're powerful vasodilators and cause permeability and exudate formation. Kinins are plasma proteins that circulate in an active form like we talked about, and then the cascade is triggered by enzymes at the site of injury, and they are also vasodilators and are chemotactic agents, and they also work through the cascade, work with prostaglandins to result in pain at the site of injury. So prostaglandins and leukotrienes are fatty acid molecules, and they pretty much do the same thing as kinins. Complement, we talked about, it's composed of plasma proteins, and it destroys pathogens when activated. Cytokines, such as TNF, alpha interleukins, and interferons are signaling molecules secreted by many cells, and they're response markers and chemical messengers. So if you do a blood draw and those are elevated, you can see that there's an inflammatory response going on. C-reactive protein, serum amyloid A, and fibrinogen are also proteins secreted during the inflammatory response, and in a blood draw can be markers for measuring a response to an inflammatory process. So all this process results in the formation of exudate, which consists of fluid and leukocytes and proteins that move from the circulation to the site of injury. The nature and quantity depends on the type and severity of the injury to the tissues involved. And sometimes the waste products fighting the infection, like pus, is also in exudate. So the key effects, to summarize, of the inflammatory response are to prevent infection, reduce damage by flooding the area and diluting toxins. The plasma proteins destroy and contain bacteria. The white blood cells, that's neutrophils and macrophages mainly, eat cells and destroy cell debris. It limits and controls the inflammatory response through an influx of plasma proteins, enzymes, and cells. It helps stage the adaptive immune response and prepares the tissues for healing. All right, well, I think that's enough for this lecture. Thank you for listening.